always thought of Rhode Island like New Jersey. It's a place you drive through to get.
But it's much prettier. People drive through it, but it's spectacular. The beaches are great history.
Okay, two actually important announcements. One is just to remind you the last session of the year we moved to June 22nd. It'll be on the faculty club. And I don't have pharma sponsoring it, so we're going to self-sponsor it. And the second thing I'm going to tell you is I'm not going to ask you to fund that dinner. We'll fund it differently. But a key thing that I'm really serious about, today is Give Big Day. The Seattle Foundation is matching donations to a lot of NGOs and nonprofits, up to $2,500. An exact match. So it's a handout. Is there a slide also, Drew, on this? Anyways, a handout back there telling you exactly how to do it. And I really would encourage you to do it. Any amount you get directed exactly into the budget that's listed here. You'll get dollar for dollar match if you give just $25 or anything. So anybody who's listening to me appreciates these sessions and wants to pay a little bit for them, I really appreciate it. It'll go into funding all the things we need for fellowship. So you can pick up this handout. You can just go to Seattle Foundation online, and the instructions are pretty clear cut. Just the key thing is make sure the money says, as I have it listed here, put it into allergy, immunology, research, and then the exact budget number that's listed, and then we get to keep every penny of it. There's no overhead for the university. So it's a pretty good deal. All right, so John had to change his date early in the year. I remember the Ebola epidemic was very hot at that time. He had to go down to the CDC for Ebola training. So now we're going to talk about somewhat similar topic, emerging infectious diseases. So thanks again for coming. This year, apparently, we haven't screwed up the broadcast yet.
We'll see.
Thank you, Len. Thank you so much for inviting me yet again. As I keep saying, you guys are always interested in whatever I have to say, so I just decide what I want to talk about every year. And fortunately, the AV system worked as well. I have a lot of slides because I get excited about this topic. Hopefully I can get through all of them. If I go too fast, please let me know. And if you have any questions, as in the past talks, the conversation is the most enjoyable part for me. So I'm happy to skip over stuff and end wherever you want. So the topic today, as Len mentioned, is emerging infectious diseases on an increasingly interconnected planet. And you can guess why I picked Godzilla there in the back, emerging from the water, heading right towards us. Quickly, I was asked no disclosures, and I have nothing. So I'm just an infectious disease doctor at the university. So there's tons of infectious diseases out there. I'm trying to, the point of this talk is to sort of build a little bit of a framework for why emerging infectious diseases are important and why we should be paying attention to them, and honestly understand why these are happening. So there's lots of reasons how you define emerging infectious disease. These are important to remember, but these are sort of some of the big blanket terms associated with this. If this is happening, this is what makes an emerging infectious disease. So obviously a new organism, the evolution of an organism, new geography spreading from one continent to another, the development of new drug resistance, which often isn't thought about as emerging infectious disease, but I hope to show you, is definitely falls into that concept. And then honestly, societal things like breakdown of public health. And honestly, societal issues underpin a lot of these, but we know that we've had resurgent infections and more difficult to treat infections as a result of changes in things like public health. So this is an interesting slide. I'm not going to go through just a little bit detail. I just think because it kind of gets across a few topics, and it's got a lot of tiny words on it. I'm sorry, but the point here is that this is from Australia, and if you look at there's a timeline of about 1800, 1900, 2000, and you look at some big concepts that were going on in Australia, right? So we went from a very, very, very rural place in Australia 200 years ago to a place that's got intermittent urbanization. And what you see here is population on this dark line going up here, right? And deforestation on these little light lines in these different areas. So these are the different states in Australia. Here's the foundation of the different cities. And then you look at the human diseases and animal diseases. And the thing here to recognize, one, we've had a lot of things happen in society in Australia. We've had a huge population boom there. 22 million doesn't seem like a lot, but for a place that had very few, it's a huge deal. Massive amounts of deforestation where there are forests. Remember, it's a very desert area. Lots of new cities being built. And what you see here is, a lot of diseases that are transmitted from human to human. So things like pertussis, mumps, smallpox, tuberculosis. A lot of animal diseases. And what you sort of see is a shift over the past 200 years into a large number of novel viruses. Of course, this is around the discovery of viruses and our ability to identify them, and that certainly has some bias. But even as you go forward into the later part of the 1900s, you're still seeing a lot of new viruses appear that fall into this big bucket of zoonotic and vector-borne diseases. So really a shift from human-to-human transmission into these sort of novel pathogens that seem to be associated with urbanization, deforestation, and increased populations. So it kind of gets across some of these basic concepts. And one of the things to recognize also is if we look across the world, about 70% of all new infectious diseases are coming from zoonotics, or basically from animals. And so these are just looking at a lot of vector-borne diseases. So where we have viruses and bacteria living in animals and being transmitted to humans via vectors. And when you're talking about 70%, obviously a large proportion of new emerging infectious diseases come from these populations. So quickly to talk about some of the other concepts beneath this, if we look at what a zoonotic infection is, it's really any infectious disease that occurs in animals and then gets into humans. And we have a few different examples of this. So for instance, if we go back here to rabies, we know that rabies is a disease that's transmitted 100% from animals. The only way you can get it from a human is to get bit by a human or to get the wound secretions into your mouth. But, if you get a rabies patient in the hospital, there's standard precautions, nothing special needs to be done. We'll skip over Ebola for a minute because this slide predated that. You go over HIV, which obviously started in animals, and it's gone through all these different cycles here, all the way up to human-to-human predominant transmission. Compared to something like Ebola, which historically was really living in animals, transmitting around animals, with occasional transmissions into humans, but really, never made it to this exclusive human-human transmission like you see with HIV. And so, basically, if you think back, rabies, Ebola, dengue, HIV-1, what you're seeing is this movement of pathogens from predominantly animal populations into sort of transient infections into humans, into predominantly infections in humans, as we see with HIVs. Far more people infected with HIV-1 than there are animals, although it's still circulating in animals. Is this making sense so far? Too fast? Okay, good.
Right.
What's that?
Just right.
Just right, perfect. I have a healthy ego, and you can tell me if too much. So here's just another conceptual way to look at it. So if we know that infectious diseases are occurring in bat populations, and we'll talk about Ebola about that, and that these infections can move from bats into, for instance, other animals like cows or pigs, right? We know that cows and pigs are domesticated animals living in a close proximity to humans. And so as long as these things stay separate, right, we're going to be okay. But as soon as these bat populations interact with our domesticated animal populations or with us, we start having problems. And you just think about all the reasons why bats, pigs, cows, and humans start interacting. It goes to those initial things I said, deforestation, urbanization, large population growth. So as we encroach into the world's forests and natural ecosystems, this is going to happen more and more and more. And that's actually what we're seeing. So I'm going to take you through a little bit about Ebola. I know you guys have heard a ton about Ebola, but I'm going to try to bring out some of the salient points and a little bit of our personal experience here in Seattle. And if this is too tedious or boring, raise your hand and I'll just skip over it. We'll take a vote. So let's go back to February 24th of last year. This is from HealthMap, healthmap.org. Anyone can bring this up. And this is sort of the status back then. There was a number of cases, mostly in West Africa. As you know, the three affected countries of Guinea, Sierra Leone, and Liberia being the big parts. But we started having cases sort of scattered out. I'm sorry, actually, this is this February. Sorry, this February. So this is where we are this year. So we had cases sort of all over the place. A year ago, February of last year, we would have never thought to see a map like this in Africa or anywhere else. Before this, there were no reported cases of Ebola in West Africa, and really no cases, obviously, in anywhere else in the world. This is basically where the big epidemic was happening, as you guys, I think, are all aware. Sierra Leone being the leader, Liberia and Guinea continuing to have bubbling along with cases right now. And the point here is, again, thinking about those concepts I brought to you. There's it's a zoonotic infection. Ebola is naturally found in bats. Which bat exactly isn't clear. They think it's largely these large fruit bats, but maybe some other bat species. And it circulates normally in these animals. So we have what's called an enzootic cycle, so a cycle of Ebola virus living in these bats all the time, not really causing them much problems. And then what happens, it escapes into other mammals when it has what's called an epizootic cycle. So in this case, you get into these little antelopes, duikers, and things like non-human primates like chimpanzees. And actually one way to detect emerging Ebola outbreak is to look for dead gorillas and chimps, because it does cause disease in them. And then you get transmission into humans. And mostly this is around hunting. In most African, at least close to or poor African villages on the edge of forests that need to hunt in order to get protein, it's those hunters interacting with animals who are butchering animals. Anybody here who butchers a deer knows that it's not a clean process. It's the same issues there. They actually, you get nicks and cuts and exposures to blood and other body fluids when you butcher an animal. And these are important protein sources for a lot of these subsistence villagers. And then so basically, if you get transmission of Ebola into one of these animals, and then a hunter accesses one of these animals, or you buy an animal from a hunter to eat, you can see what can happen. And so we know this is the process that's probably driven all the Ebola outbreaks that we know about for the last 40 years. So, as I mentioned, we never had an Ebola outbreak in West Africa. We've always had them in basically Uganda and the DRC, the Democratic Republic of Congo, and Sudan, a couple other places. Really, this dense area here in the middle. This map is actually the fruit bat, we think is the main animal that harbors Ebola, is in this middle part of Africa. So this totally makes sense. This is where most of the outbreaks have been. This is where the bats are. But, people haven't really paid attention to this upper left corner right here, where there's a lot of these bats. This is a large, large area. And obviously this overlaps very nicely with Liberia, Sierra Leone, and Guinea. So actually for many years, people have been reporting Ebola antibodies in bats in these areas, but no one's ever detected an Ebola outbreak. But it makes complete sense in retrospect that there was an Ebola outbreak coming here at some point. We know we had the animals. We know we have the type of subsistence living that leads to that hunting and so forth. And so, again, in retrospect, not a big surprise.
John, wasn't the index case in this most recent outbreak a child?
Yes, I'm going to get to that exactly. We're going to get to the child here in a moment.
I see, because it like that model of going hunting and subsistence. I was just curious if there was, maybe you're going to talk about that.
Yeah, I'm going to talk about that explicitly. So here's the bats. We know it's the distribution in Central and West Africa. Here's actually the distribution of those bats globally, right? So that's the dense areas, but they actually spread pretty far. So again, when we think about why diseases, infectious diseases emerge, we have to think about an association with animals that harbor these viruses. And we have to think about the spread of these. So that's where these bats live. If these are really the bats that are the drivers of Ebola infection, we have a much larger area to think about, including some incredibly populous areas of the world.
Do they see Ebola in the bats in that whole region?
None reported yet, no. But we'll come back to something like H5n1, which is spreading pretty quickly and is now in the United States. I'm not trying to be, trying to paint a nightmare picture here, but we need to think about it at least, be aware. That's why these concepts, I think, are important.
Since you're showing such wide geography, I've always wondered why just those three countries, it must be equally poor with the same practices just adjacent to them. Why did it stay in those boundaries?
Well, it did. It went to Nigeria, it went to Senegal, it went to Mali, but, it's not actually an easy disease to transmit. It's just not. It's actually pretty hard. And I mean, I can get into the biology of it, but basically you need contact. It's a disease that preys on families. So at close contact, you get someone who gets sick, right? And you, basically in that early phase of infection, they're not very contagious when they're sort of just feeling ill. As soon as they start getting sicker, the viral load starts increasing, and the number of body compartments that it spreads to increases. And so as you get sicker and sicker, you become more infectious. That's when your family comes in and tries to take care of you in a place that doesn't have a health system to take care of you and isolate you. And so the family members actually start taking care of people when they become the most infectious. Then they get infected, and then they may feel okay. They stay in that place, may go back to their homes, and then their family members take care of them when they get very sick. So it's not like these people are out in the community spreading Ebola. It's really a very specific contact that leads to transmission. And so these families aren't crossing borders. We were worried about that because the borders here are quite porous and sometimes separated by nothing more than a river. One village on this side and a village on the other side. And there was probably cases that went undetected that were probably just across, but it was not something that spreads like that. The big driver, and we can certainly talk about it as an aside, of the difference in the Ebola epidemic in West Africa was the urban component. We've never had an urban Ebola outbreak ever. There's always been small outbreaks in villages that have been pretty isolated, detected fairly late after everyone is sort of either infected or died. And so you come in and then you save the world. Actually, you didn't do anything, right? It's already done. Either you're infected and lived or you're infected and died. But you couldn't get out of that isolated area. The difference here is that people are moving back and forth in urban areas and rural areas. And so you have kids going back to take care of grandma who now is sick and then feeling better and going back to their urban area, to a slum area, and then getting sick, and then sister comes and takes care of that person who's now sick in an urban area with no health care. And then you start seeing it spread in those settings. It's something we can spend the entire time talking about. So as I mentioned, this is the slide I thought I was talking about before. So here's go back to March 14th last year. This is when things first started being detected by Doctors Without Borders. They were notifying the world. No one was really paying much attention. Sort of these outbreaks up in Guinea. And epidemiological studies, as pointed out, have sort of honed in on actually where this all started. So we're in this place called, here's along the road to Gekudu up here. And I think this is the kind of the area up here. So there's an anthropological investigation up in these areas looking for where these point outbreaks were. They did some bat capturing, biomonitoring sites. This is actually the village that they brought this down to, that they think where this happened. This is the house of the index case, thought to be a 2-year-old boy. And this is actually where they think he actually got infected. So this tree, the boy from this house went to this tree and got infected by a bat. And this is where the Ebola outbreak that we've all been dealing or listening to or acting in for the last year started. This is his village, and as pointed out, this wasn't a subsistence hunting situation. This is actually, a fairly okay village. We got metal roofs, is a good sign, and but folks probably using the forest for resources, but also playing. And this was a tree that was a little bit offside the side of the village, and a lot of kids liked to play in. They would go up there and hide in this. And what happened was that the kids, this little boy would go up there with his friends and they'd play in there, and we know that this thing was full of bats. Not the big fruit bats, another type of bat, which they think may have Ebola. Unfortunately, since this all happened, the villagers went back to this tree and burned it. But they were able to, so all the bats are gone, obviously. But they actually found DNA from the bats in this tree, so they know that they were living there. And so the kids would capture bats and play with them, these little bats, and handle them, and like you catch a snake or a turtle. And Ebola moved from that bat into this child, and then from that child who died to his sister who died to his parents and so forth, and just spread from there. And family members came in to take care of sick relatives. This happened over and over again. But there was subsistence hunting there as well, and so this is a typical bat hunting instrument. So you can imagine what happens when you go up to that tree and you jam this thing up in there, and you retrieve a big bloody bat. How kind of messy. This is a very effective tool for doing that, but also an effective tool for bloodletting and transmission of zoonotic pathogens. So it's fascinating to me that they're able to. This is the tree where this whole thing started. Really amazing.
The bats don't get sick.
The bats don't get sick.
Why?
Well, that's the classic issue of a zoonotic infection, right? So, or pathogen is that there has to be an animal that harbors it without disease. You know, maybe it's something like HSV in us, you know, occasionally makes us feel cold, but mostly it's fine. Or CMV or EBV. You know, we harbor lots of things that we don't have any problems with. SIV, immunodeficiency virus, and pseudomandibes, they're huge viral loads. No disease, no problems. They live just as long as non-infected pseudomandibes. So that's how it works. That's an epizootic and zootic cycle. So this obviously built up. This is October of last year. I think everyone knows how continued to transmit. We had cases in the United States. We had lots of media, which I could spend the entire time talking about, but I'm not, because I will get ahead. You know, lots of media, people wearing all kinds of stuff, levels of protective gear. You know, who thought that we'd be wearing spacesuits like this in the hospital? But also, just kind of interesting as an aside, you know, this is what the CDC said. You know, this is Ebola. You just need to wear this stuff and you're fine. And not one hospital in the country kind of believed that, except for the one in Dallas. And yeah, just craziness. We built up whole rooms. So at Harborview, we have an emergency room. We have hot rooms and cold rooms. I know more about hot rooms and cold rooms and Ebola pathogens than almost any other infectious disease now. And decon, and this is upstairs. We've had an isolated ICU room now for the last 7 months, just for this purpose, for rule-outs and so forth, where we have, this is one ICU room that's been isolated that no patients have been in 7 months aside from Ebola rule-outs. We have warm rooms, doffing, donning, colds, everything built. We built showers, everything. It's really changed the way we're doing a lot of infection control in the hospital. This is just pathways. We've had to sketch out so much stuff. It's unbelievable. So I just wanted to make sort of an aside also. So here's U.S. national approach. I think in your slides there's no question mark. So there was this active post-arrival monitoring of travelers from Liberia, Sierra Leone, and Guinea. How many cases have they caught using this? Zero. Huge waste of time and money. Huge. They've caught zero. Nothing. No suspect cases. All this screening. No. There's enhanced guidance from the CDC. How many people followed that? Zero. At the beginning. They got much better. You know, trained observer, you know, the response team, zero. I mean,
when you showed us that diagram of the new way to isolate these patients, has that been adopted by all the hospitals that want to take care of these folks?
For treatment hospitals. Now there's a tiered hospital approach. There's a frontline hospital, assessment hospital, and a treatment hospital. So in Washington State, there's 3 treatment hospitals. Seattle Children's, Spokane, Sacred Heart in Spokane, and Harborview. So those 3 hospitals have this. If you're an assessment hospital, you don't need to be at this level. You need to have you have to have a preparation that's similar to this, and you need to be able to take care of a patient for up to 3 days. If you're a treatment hospital, you have to be able to take care of a patient for up to 3 weeks, and you need to have very high level this. So to get this status, we were visited by the CDC assessment team, and they went to Children's and Sacred Heart. So there's about probably 50 hospitals in the country that can do this. Yeah, huge amount of work. But the point here is that, you know, we have this picture of disaster responsiveness and ability to handle things, and actually it was completely uncoordinated. It wasn't well done, actually. I think we all might have that impression. And so hopefully we can learn from this, because what, you know, national authorities were saying about isolation screening, how to take care of patients, and what hospitals are actually doing on the ground were very, very different. So again, let's bring it back to, am I putting you guys to sleep? You okay? Okay, good. Okay, so let's talk a little again back to the concept. So I sort of talked a little bit about Ebola and this transmission. So we know that, again, this is the point. Look, here's a bunch of animals that can have viruses or other infectious diseases, and they, you know, sometimes have disease and sometimes don't. You have little epidemics that occur in their populations where they just kill off or they just live with them. We have vectors up here, which obviously can play a role in transmission. And sometimes you get spillover from these wild animals into domesticated animals. Cows, pigs, ducks, camels, as we'll come back to. So that's this spillover phenomenon. There's actually a great book by David Quammen called, I think it's called This Spillover, right? What's that?
It's an amazing book. Gary did this talk, a year ago, Gary, after reading that book?
Should I just stop now?
No, we forgot all that.
Well, feel free to comment on this. It's a very good book, very good, well written. He writes good stuff. I think it's called Spillover.
Yeah.
Anyway, so you get the spillover from animals into domesticated animals. Sometimes you can get it directly, like in the case of these bats, into humans. And then obviously within the domesticated animals, you can get spillover into humans. And you can get amplification within the domesticated populations, and obviously amplification, like we did with Ebola in the human situation. But the point is that there's lots of things that can be done if we know why these things are happening to curtail this. So forecast readiness. Have you guys heard of viral forecasting? Basically, the idea is that we do go out there and look at what's happening in these hunter-gatherer populations, what's happening in people living at the edge of forests and so forth, where a lot of these things are coming out. Farmers in large domesticated operations in South Asia, for instance, where we see a lot of influenza viruses, or hunter-gatherers. And actually, there's a guy named Peter Rabinowitz at the UW who is active in this field, and this actually does like testing of veterinarians who are doing research in Africa, and we'll get blood spots and test them for like every possible virus. Really cool stuff. So that's looking at what's possibly out there, early detection. So diagnosing people, you need a health system to do that. And then there's obviously things you can do. If you know things are coming, you can do something about it before it gets into animal populations that are living next to humans, and before it gets into humans. So you can kind of blunt these huge, agricultural effects and big human effects by having processes in place to diagnose or forecast, diagnose, treat, isolate this. And to be honest, we could have done this with the Ebola epidemic, and it was missed on many, many, many opportunities. The Ebola epidemic never should have gone as far as it did, and it was really a failure of international public health on many levels. So quickly, let's talk quickly about influenza. You guys know all about influenza. We know that.
On to that.
Yep.
Why did this epidemic now end?
Well, so I think that we know how Ebola is transmitted, right? It has to do with identification, isolation, contact tracing. It's a classic public health mechanism. We know how to do those things. The fact was that they weren't done. And so we have a place where you have no health system, right, where people don't have access. And then you go in, and they're sort of, it's out of control. Things get quickly out of control. By the time we come in, we've sort of missed these opportunities to blunt this. So we're dealing with this big red spike. But most infectious diseases eventually, peter out. You educate the public. They start learning what needs to happen. The place where it happened the fastest was Liberia, where the U.S. pumped in a bunch of money and actually did that contact tracing. So you provide quality health care for people where they're not afraid or less afraid to put their loved ones in healthcare and be isolated. Because for most people, the first 9/10 of this outbreak, your loved one went into one of these places, they never came out, except in a body bag, and you weren't allowed to touch them, right? And so there was also this idea of taking, bodies were being put away in places that emphasize the care of people who recently died. So the bodies, handling the bodies, which are the most infectious. A dead body with Ebola is the most infectious thing that and if you're handling it, that's when you get a lot of transmission. And so it was education, getting people to the right burials, contact tracing, and trying to save people, and honestly learning how to treat people. This was actually a big. It was more like cholera. We think about Ebola as a hemorrhagic disease, and it's a small thing. These people were losing 9 to 10 liters of stool a day. They were very, very ill, but they were dying because of dehydration. So electrolyte abnormalities, renal failure, not bleeding out. It's not like, on the movies. That, the coagulopathy was certainly a part of it. But you get these people in and you pump them full of fluids early. They do much better. So, you start getting better outcomes. And we've learned a lot from that from the North American experience. I mean, look at Nina Pham and Amber Minson, the two young nurses who were infected. Now, great, they had youth on their side, in their 20s, but, Nina Pham, she sailed through. I mean, I don't think she ever got very sick. And I don't think Amber Vincent did either. So early interventions, learning how the pathogenesis is occurring, and honestly, communicating, educating the populace so that they will engage in care. If they're hiding from care, that's what's going to amplify the epidemic.
How long will the virus stay in a dead body alive?
I don't think we know. A long time. As long as there's organic matter there, I think a long time.
Because they're worried now about the sexual transmission of it, and they'll 3 to 6 months, the residents.
Yeah, I think that.
How does it, like Lynn's question, how does this whole thing stop? Why doesn't it become like HIV?
Well, I think that's the big question. I don't think we know yet whether it potentially become. I think a big concern is whether it can potentially be an endemic thing. Are we ever going to see no cases in Guinea and Sierra Leone? Liberia, we're looking really good right now, but are there going to be more cases? Is it going to be more kind of a low-level endemic infection? It's hard to know. The difference between this and HIV is that it makes you very, very sick, very, very soon.
Are there any people that have gotten it a second time? In other words, do they have lifelong immunity, it looks like, or how long?
It looks like immunity. It's unknown whether it's lifelong. It looks like, because they use a lot of Ebola survivors to take care of Ebola patients in many of these countries. Very effective. And there was no known bad outcomes for Ebola survivors in these populations. So it looks like, and we know the vaccines work, right? So unlike HIV, the vaccines work that we have. So we can develop good immunity, but whether it's how long it lasts, right? We're still learning things about the pertussis vaccine and other vaccines about how durable those immunity.
What do they look at? Maybe 50% fatality, and then you have all that 50% that survive. That helps prevent epidemics
Yeah,
as well.
no, it's a good point. But you remember that in there, fatality was 50, depending where you are, 75%, 80%. Just as an aside, in the United States, we have that one guy, the unfortunate case in Dallas, who was quite sick when he came in. And we had the physician who was transported over, who was also quite ill. Everyone else lived. So even some of the cases in Emory were extremely ill, intubated, on dialysis, and we still got they got them through. And so the fatality rate for treated Ebola is actually probably 20%, maybe even less. So if you detect it early, treatment is very effective. But that's just an aside. But the vaccines work, so very different from that. And whether we can ever use wide-scale vaccination in these populations is probably it. So if you think, if it's a zoonotic infection, who do you vaccinate? Right? You vaccinate everyone where there are these bats. That's a pretty big area. It's possible, but it's different than HIV. We're not worried about new HIV coming out of primates, not human primates. We're worried about Ebola coming out of bats and other animals. It's just a different pattern. This is exactly the thing that I wanted to talk about today, because I think that's what's interesting. If you know about the patterns, that informs what to do about them.
How do these viruses differ so much that it's easy to get good immunity for Ebola? It's been impossible to get a vaccine for HIV.
Well, the thing about this, it's amazing about HIV, is that at every level of the immune response, and there's some guys, I'm sitting in a room full of immunologists, it's a little daunting, but when I did my research in HIV, it's every level of immune response, from the intracellular sort of innate immune response to the more complex, T cell and B cell responses. HIV circumvents almost every single step of it. So it's my hypothesis. HIV has been coming into the human population for thousands and thousands of years in small little outbreaks, because it's so well adapted to bypassing so many parts of our immune response, as opposed to these viruses which infect other cells in our body, right? Ebola doesn't deal with T cell infections. But HIV, it targets the immune response and is able to also generate so much, it mutates so rapidly as part of its normal replication. It's able to bypass that adaptive immune response in a way that Ebola is not. Even though Ebola is an RNA virus, it's still pretty messy, but not nearly at the level of HIV. Is that, for the immunologist, is that rational? Mm-hmm. Okay.
Speaking of that, drifting and shifting.
Yeah, so quickly, this is just an intro. I know you guys already know this. Influenza A is the big drifter and shifter versus influenza B. B is the one that floats around in humans, just as a reminder, so that's why we don't have big problems. It only infects humans. Going back to our zoonotic issue, these are my drawings. Do you like them?
I'm sure your kids didn't draw that.
Come on.
I saw you in a gallery downtown.
Yeah. So the issue is we never talk about influenza B as being this big, dangerous virus, because it only shifts around in humans, right? And we have only that drift. The issue with influenza A is that it goes into ducks and pigs and other animals, and when you get these mixing vessels, we get big shifts, right? And it's because it's a zoonotic infection. So that's flu A. We've had a ton of pandemics. Most recently, the 2009 H1n1 pandemic. But we've had, which caused huge amounts of disease. And I'm not going to go through the kind of the epi potential of the 1918 epidemic that occurred now. It's certainly possible. For those of you who are taking care of patients, this was a huge widespread outbreak that started actually in Mexico. We were all looking at, Asia as a place that this pop out. We totally missed it. This came out of Mexico, and this was a recombined virus. So we had a combination of Eurasian swine, classic swine H1n1, human H3N2, and avian influenza that mixed together into these North American swine H3N2s and H1N2s that recombined again to generate the 2009 H1N1. So that's shift. That's shift. When you're having these recombinations in ducks and pigs and humans, we have this outbreak. And that's why we have pandemics in influenza. This is what happens. We have no prior experience with it, no natural immunity to this or ability. And so it's a brand new virus that emerges out because of our association with animals. And things like this. But this gives me the opportunity to shift over. So this is, when we think about the agriculture and how we deal with animals, so this is a more traditional, if you go back, 100 years or 200 years, this is how you deal with pigs. They're out, they're hutches, and they're walking around. But things have changed, right? And I'll just be clear, I eat meat, so I'm not dissing anything here. But this is much more like the way we deal with animals now, right? This is large-scale industrial agriculture. Lots and lots of pigs. And if you're thinking about how to transmit infectious diseases that start in pigs, this is the way to do it. And we have human beings in those places. So, as an aside, who do you think the company that owns this cares the most about? The pigs, right? This guy's replaceable. Whoops. Unfortunately, these animals, they're the ones that they're the money. And so but the important thing is we have humans in these settings who have the potential to infect the pigs and have the potential for the pigs to infect the humans. So here's just a slide. You never thought I'd talk about hog operations in the United States when I came here. Sorry, never going to get invited back again. But what's cool is that here's the dark line is the total number of swine operations in the United States, going from 1965 to 2009. So what you're seeing is a huge decrease in the number of swine operations in the United States. At the same time, the average number of pigs per operation skyrocketed. So what you're seeing is the, basically the industrialization of pig operations. So fewer operators. This is not small mom and pop pig farms anymore. It is big factory farms with thousands and thousands and thousands of animals. And again, thinking about what I told you about how zoonotic infections and emerging infectious diseases occur, this is something that we have to really pay attention to. And the other issue is that this is not happening just in North America. So that's in the orange, where it's really skyrocketing, are in developing countries. So again, thinking about the drivers of zoonotic infections or emerging infectious diseases, we're putting in new accelerators. Remember those curves where you can accelerate, amplify infections? Here's a perfect example of that. So here's just an example of how we think the H1n1 virus may have occurred. So we have bird flu, human flu, swine flu. We have a swine worker. You can just think about this. I'm not saying this is perfect, but here's one idea. You have a swine worker who's maybe infecting a pig. The pig transmits back to a swine worker. Swine worker then goes home, starts transmitting human to human. I mean, this is a classic sort of issue. And when you have tons and tons of pigs, you can imagine how the mixing can occur. Not really a stretch of imagination. So again, here's looking at birds, millions and millions of birds all floating together. And here's just the chicken population in the United States. That's in thousands. So you can do the math. Add 3 more zeros to the 20 million there. Trillion? Yeah. Huge numbers of animals. And so this brings us to H5n1. So everyone's heard about H5n1. The avian influenza has affected about 600 so many people in the world. Mortality pretty high, 50 to 60%. but not easy to get around. We have pulmonologists here in the audience. The thought is that the sialic acid receptors for H5N1 are deeper in the lungs, so it's actually, once you get infected, it can cause a very serious pneumonia, but it's actually pretty hard to transmit because it doesn't attach in the upper airways like a lot of other influenza viruses do. But aside from the pathogenesis, H5N1 is now in the United States. It's in birds. The risk to humans is extremely low, but it has caused a lot of problems. People have been paying attention. This is the viral forecast, seeing what's coming across the border. So 694 infections, 402 deaths, so the mortality rate is quite high. And you can see it started in over here in Asia, and it's sort of spreading this direction. So when you think about why it's here, we have to think about the birds. Again, I talked about pig operations. Well, here's bird flyways. And so we know that those infections started out here, right? And they're sort of trending this way. But when you think about the birds that can carry these infectious diseases like H5N1, you have to think about where they're traveling to. Like I showed you the bat map. This is a flyway map. So this is where they migrate to. So they may spend their summers, in one part of the world and the winters in a different part of the world. And so if you think about all the birds in this part of the world, look where this blue line goes and where it ends up over here. So actually some of our biggest forecasting is up in Alaska because a lot of these birds fly all through Asia, all the way up to Alaska, and then fly all the way back. And then you have overlap with large numbers of animals in the green lines here of birds starting up there, maybe nesting in the same place, and then traveling all the way down, the west coast. So it's not a big surprise, again, to think about how this would happen. So I have 15 minutes left. I'm going to keep boogieing. Is that okay?
Yeah.
Everyone awake? Here's just another outbreak, H7n9. This is an outbreak that sort of is mostly petered out. About 140 cases. This is all in China. What's interesting about this is it's yet another influenza virus, not super widespread. Fairly high mortality, 45 deaths and 140 people in China. One was China did a great job of detecting this and talking about it. They've learned a lot from SARS, and they've done a really great job. But the thing that really was remarkable to me is that look how big this area is. This is all of eastern China. And there are cases throughout eastern China detected. Now, part of that is a laboratory detection bias, a diagnosis bias. But they were able, in a very short amount of time, a few months, find these cases throughout eastern China. Really remarkable that this spread so quickly, but fortunately petered out. Here's just the histogram. So they have an early bump in 2013 and another big bump in 2014. A lot of people who don't pay attention didn't hear a lot about it, but this was a new, novel. We never think about 7s and 9s in humans. Every year, I try to track how many H's and N's, neuraminidase and hemagglutinins, I need to pay attention to. I've stopped doing it because there's just too many added. There used to be like 3 or 4 hemagglutinins and 3 or 2 neuraminidase. I can't keep track of it anymore. So again, let's switch over to humans. So this is the way many parts of the world live. Here's a woman who's getting her washing water, her drinking water from the same place that all these domesticated cattle, are drinking and defecating and urinating and washing in as well. This is a pretty common situation. So if you think about transmission, not a big surprise. But here in the United States, where we have those large industrial sites, what do animals make? Stool. They make a huge amount of feces. And so if you go to a large industrial agricultural site, it's common to have sewer ponds, basically fecal ponds. There's no place to put all this stuff, and you make an enormous amount of waste. And so they just make these big ponds. And this is just full of feces. And so you can imagine the stuff that's brewing there. And you can imagine that if you're farming right there, this is not concrete lined lake. Stuff is going to move. Wind's going to come over this and blow stuff right into those areas. And as an aside, if you think about the number of antibiotics the animals get, and antibiotics in there, mixing with all those gram-negative rods and stuff, big problem.
Has this changed as they started using the fecal particles in the irrigation when they irrigate the plants? They just literally flush all that into the system and pump it out onto the crops.
Yeah, I know a little bit less about that. I've heard about it, but I'm not enough to talk about it. I'm sure it's not great.
Is the virus in feces or is it airborne?
Influenza virus?
Yeah.
I don't
is this really a risk for?
So I'm jumping from influenza to other stuff, back to zoonotics. I'm sorry. The point here is that you think about this, and you think about antibiotic use. Remember I talked about emerging in antibiotic resistance? Antibiotic resistance as a driver of emergence. So for many, many years since antibiotics were discovered, farmers very early on recognized they did two things. One, if you have big industrialized situations, they have lots of animals close together, eating foods that they're not used to, having diarrhea all the time, getting ulcers, getting stressed, giving them antibiotics keeps them alive, right? So that's what you value. That's reasonable. But they also recognize if you give them low levels of antibiotics, they grow faster, fatter. And so this was, just the old, advertisements giving antibiotics to animals. And this is still very, very common and pretty much uncontrolled. There are lots of human classes of antibiotics that are using animals, not only to prevent them from getting sick, but also to get them to grow faster. If you think about that and the number of antibiotics they get, you're talking about the perfect test tube for generating antibiotic resistance. And if we have people going back and forth in those settings, you can imagine that being a big problem. I'm going to come back to that. Anybody see that movie, Contagion? No? No infectious disease doctors. It's like a great infectious disease movie. Yes.
When you cross into Canada now, they take all your eggs and dispose of them. They won't take eggs across the border anymore.
Wow.
Because of the avian flu question in Canada.
Yeah.
Well, and then wasn't it just last week Tyson?
Okay, let's...
Tyson, which is the biggest chicken producer, no longer is going to do antibiotics.
That's huge. No antibiotic classes they're using in humans. They're still going to use antibiotics.
Yeah, they'll use them, but not the ones that humans
Yeah, and I think that the consumers are becoming more aware of this. How many chemicals do you want in your food? I don't think consumers are necessarily aware of the antibiotic resistance issue, but I think they're worried about chemicals, and certainly they're getting a lot of pressure from the government. And I think this is coming up and up and up, and I'll talk a little bit about some of the governmental pressure in a moment. So I'm going to quickly go through the rest of this in the next 10 minutes, okay? This is, I forgot her name, she's an actress. Gwyneth Paltrow. She was in a movie called Contagion, and it was about this infectious disease that she got from contact with a farmer who hadn't washed his hands, a cook who hadn't washed his hands. The point is, it was this horrible meningoencephalitis that was transmitted by respiratory droplets and so forth. And so it's this horrible thing. It's not a virus that really exists, but it's based on a real virus called Nipah virus. And this was a big outbreak that happened in Malaysia back in the 1990s. And the point here is that one, it was a disease which no one had described before. But two, it's spread now. It was a very dangerous disease. It does cause meningoencephalitis, but it's not transmitted by respiratory droplets, so not as bad as the movie, but a real disease. It's now spread throughout this area and into North Australia. But the other thing is the way it occurred. The reason it occurred was because it was another bat virus, Nipah, and these Hendra and these other viruses are from bats. Is that the people in Malaysia would build pig sties and make a big thing with roofs, rafters, and so forth. And then to add on, they would burn everything around it and then put plantations up with fruit trees. They brought in fruit bats. Fruit bats would feed on the trees, go up and roost in the pig sties. They're very messy eaters, so they would chew, and spill. The pigs would then eat this stuff. The pigs would then get Nipah virus. The people would then interact with the pigs, and they would get Nipah virus. So this actually had a huge deal because they had to basically kill most of the pigs in Malaysia, which devastated the economy for these folks, as well as the horrible medical outcomes. But now, widespread. So again, these interactions of deforestation, commercial farming, bats, pigs, humans, all together. And then you get a really cool movie out of it. If you guys haven't seen it, you get this. So deforestation, I'm not going to spend much time on this. It's a big deal. I think I've convinced you of that. This is pretty common to go and burn a forest down to provide more land for farming. We think about how much this is happening. You can see this is net loss and net gain. So net loss is up here. And we all know about South America and the loss of the Amazon. This has happened hugely in Australia and through the Indian Ocean, parts of Africa. We've seen reforestation in places like China and North America. But that's not also, when we say reforestation, it's not replacing the forest, right? We know much of the reforestation in our state, Washington State, is monoculture. You go to a reforested area, it's the same tree, right, planted a million times. And so there's actually been data to support that as a problem in and of itself. This is looking at land cover change and looking at the number of emerging infectious diseases associated with those land cover changes. And actually, if you just replace that deforested area with same trees over and over again, there's actually an association, not causation, but association with more emerging infectious diseases because you've still displaced the natural ecosystem there. So you can't just go in and just, put a lot of pine trees in and expect things to be that different. So quickly about you guys, we got the little kid licking pigs. People love their camels. I guess they can be friendly animals. We know about MERS. It's going to be a problem. This is spread. Something we talk about all the time. I'm going to skip that part for the sake of it. We've had a live transmission, including nosocomial transmission of MERS. Again, this is coming. It appears to be pretty clear that camels are the vector for this. This is at least the report showing that they're able to find antibodies to MERS in camels. I'm going to go quickly through this. We need to think about animals. We need to think about diseases in a much broader context. Human health, animal health, environmental health. Unless we start thinking this way, zoonotic infections will continue to be a serious problem for us. This is now embodied as the One Health concept, if you haven't heard of it. I hope you'll hear more about it in the near future. But we need to be talking to veterinarians. We need to be talking to our environmental colleagues under one big umbrella. I'm not going to go through all these, but we all influence each other. When we talk about urbanization, deforestation, industrial animal agriculture, what happens in our forests and so forth and our humans is really important. I'm going to skip this part because I want to give time to chat, but you can look through this as cases. Here's a nice interesting one. Chikungunya is now, endemic in the Caribbean. This appears, we thought it was a reintroduction, but it appears to actually be something that pops up every 50 or some odd years. Now it's back, getting into Florida, and the vectors for these extend pretty widely, which you can see there. We've had effective responses. Again, I'm going to skip through this. This is West Nile, some disease we didn't know about. Again, introduction from different continent. And here's a whole bunch more of these encephalitis viruses. Heartland virus is described in the last 2 years. A few cases, but who knows what the future holds. Here's the factors, which you guys can certainly read about. I'm not going to go through all of them. They're all the things I talked about. Things like disasters can certainly cause big problems, not surprisingly. There's a huge cholera outbreak, and then the increased risk of malaria in Haiti after the earthquake. And then what you mentioned, the government's paid more attention to this. I think our countries worried about antibiotic resistance, and this is a report that came out in 2013, and these are the list of threats. So even though none of these are zoonotic infections, these are all antibiotic resistant. So when we talk about emerging infectious diseases, antibiotic resistant bacteria are certainly a big concern. Everyone's worried about MRSA, but we've seen the carbapenemase resistant Enterobacteriaceae, CREs, associated with duodenoscopes in the paper. That's becoming a bigger and bigger problem, and it affects things like gonorrhea, which continues to develop high-level resistance for very common infection that people don't like to take antibiotics for. We misuse antibiotics a lot. Again, sorry, I'm whipping through these because I want to leave time for questions. I want to just leave you with a couple points here. This is a really cool study where they went up to the Yukon up here outside Dawson City. You know how people do those ice plugs to look at changes in the environment? They did a tundra plug, and they looked at the bacterial DNA that existed at 30,000 years ago, and they did this very hygienically, kind of sterilized everything around it, did this plug. And they were able to find in bacteria from 30,000 years ago the genes BAN A and BAN B. BAN A and BAN B are the genes that can give vancomycin resistance to enterococci. So BRE, vancomycin enterococci, those genes are already there before we started treating people. This isn't a de novo gene. Bacteria and fungi have been fighting each other for a billion years. We're latecomers to this. The antibiotics we use are all derived from them. So we're using their tools to try to fight them off, but they already have the mechanisms to resist this. I'm not going to go through this very carefully, but this is actually this really cool shotgun metagenome analysis of the Yanomami people who have been unexposed to healthcare, actually outsiders. This was just published this year, looking at different mechanisms of drug resistance, beta-lactamases, chloramphenicol resistance, penicillin binding proteins. These people had never seen antibiotics, never been involved in healthcare, and they found in both their fecal and oral flora bacteria that harbored mechanisms of drug resistance in them already. So when we start thinking about applying antibiotics to agriculture or humans, the bacteria have already figured out everything we could possibly do to treat them. And so if you misuse antibiotics, we're going to create more resistance. It's inevitable. And antibiotics are being used widely. Most people under 5 who die of infectious disease die without antibiotics. So it's getting to the people who need it and keeping it away from the people who don't. And again, I'm going to skip all this stuff. This is a recent publication showing a whole, you can see there's 20 patients on there infected with a colistin-resistant Acinetobacter baumannii infection. So colistin is our last go-to detergent drug for highly drug-resistant pathogens like Acinetobacter. These guys are all resistant to our last-ditch drug. Very concerning. And I'm going to close on this because I'm actually close to 8 o'clock. Remember, Pit Against Microbial Genes, we have Manly Arwitz, that's Joshua Gutenberg, the Nobel Prize winner. I'm happy to take questions. Sorry to go so fast towards the end, but hopefully you learned something. Thank you. As I told you, I get too excited about this. I actually deleted a lot of slides.
Well, you're not going to have to worry about permanent employment.
Oh, no. No, I don't think so. I think we've got the, yeah. There's diseases that are dealing with now that didn't exist, you know, a year ago, 2 years ago.
So we were talking earlier before you started about Marv Turk, and I remember Marv teaching me that the biggest improvement in infectious disease was the epidemiology with tuberculosis at the turn of the last century, just how we changed living conditions. That did more than anybody in infectious disease.
Mm-hmm.
I don't know if that was a true statement, but obviously this is a completely different ballgame.
Well, certainly. I mean, go back to that first slide, the Australia experience, where we had a lot of human pathogens. So syphilis killed tons of tuberculosis, tons of people. You know, strep pyogenes, tons of people. But we're you know, where we have healthcare, or good healthcare, you know, TB is not really a big a problem. We lose about 1 million people a year globally to tuberculosis. That's much better than it used to be, and it's getting better. But the big problem with TB is not TB, it's drug-resistant TB. So MDR TB has the risk to eclipse regular TB as the major killer in tuberculosis. But we've had huge gains in tuberculosis. Syphilis, where we have access to healthcare, huge gains. Where we don't, still very, very problematic. But you're right, things like sanitation have certainly, you know, changed the face of this. But other things too. I mean, certainly treatment for tuberculosis. You know, people were commonly treated in North America for TB through the 60s, 70s. And now we're pretty effective. Most people who have TB in this country came from a place where it's endemic. So I think the issue is, you know, we need to think about everyone is connected. As I mentioned, at the beginning of the title, this is this increasingly interconnected situation. And this kid, who I took care of, David Ricci, got his leg ripped off in India while he was volunteering at an AIDS orphanage. Gets taken care of there, shows up in my hospital in Seattle with these pathogens growing out of a giant wound in his leg. You can see all those bars, right? So I have to think about antibiotic use in India leading to this. And this stuff you can find in the water and the side of the streets in metropolitan areas in India, and showing up where I live, and leading to this. You know, this isn't everyone else. This is what's happening in our country. This is de novo, right? This isn't transmitted from some other country. But this can now get to another country and transmit from hospital to hospital. So it's a long-winded answer. But you're right. Marv's right. But I think we have to open our eyes up to what's the bigger picture. Recognize that we're transitioning from smallpox, TB, to all these new things. And we're certainly making more problems.
Before people leave, don't forget, today is the only day you can get this matched donation. So please do it. We don't have enough money in this division to pay everybody's salary.
Frightening world out there. Like doing a fecal microbiome test.